of course, first of all, Barbara and the foundation, as well as CIHR, it's a real honor to get this award. And I really appreciate both, of course, the support from my lab and also just the general support for basic research over the years. I guess 10 years now. It's really appreciated in the community, and it really, I think, means a lot to everybody here particularly. So I'm going to talk today about work that is done in my laboratory, studying the neural network that generates locomotor activity or walking in mammals. Okay, so this is known, and I'm going to be referring to it in my talk today, as the locomotor CPG, okay, or central pattern generator. And I actually use a lot of similar techniques to those introduced by Rob Brownstone, the first speaker. So it's really nice you guys have kind of been warmed up to a lot of stuff I'm going to be talking about today. So the locomotor central pattern generator, or basically all of the hardware that's required for walking, is located in the spinal cord. Okay, so the most visually striking evidence of this comes from this video that I'm going to, actually I didn't check beforehand, but I'm quite certain will work. This was an experiment done in Brian Schmidt's lab at the University of Manitoba, and what he's got here is he isolated the spinal cord with the hind limbs attached of a neonatal rodent. Okay, the tail is down here, it's pinned in this dish. Okay, it's kept alive with artificial cerebrospinal fluid that's being oxygenated, and when you add drug agonists, or agonists to specific neurotransmitters, what you get is this alternation of the left and right hind limbs in a behavior that looks very similar to walking or locomotion. Okay, so this is basically just proof that all of the hardware required for walking is located in the spinal cord. Not always is known that it's located in the spinal cord, but that same laboratory, Brian Schmidt's laboratory, as well as the laboratory of Ole Keene, have shown that it's concentrated, the neurons that make up this central pattern generator are concentrated in the ventromedial regions, so the regions close to the central canals of the thoracic and lower thoracic and lumbar spinal cord. Okay, so here we've got the spinal cord, the regions in red contain most of the lymphogenic neurons, the neurons that are involved in generating locomotor activity. Okay, so even though it's been known for almost a century that all of the hardware is in fact located within the spinal cord that generates basic locomotor rhythm, and for at least the last 50 years that it's located in the ventromedial regions of the spinal cord, the identification of the components of this neural circuit as well as the manner in which they're connected, so its basic structure and function, haven't really been that well described. And the reason for this is very similar. It's funny that Rob had almost an exact same picture as this, except it wasn't in color. Not that that's better or worse. The main issues with studying this neural circuit is exactly what Bob said, the problems with studying hand function are. This is a 20 micron thick section cut from a neonatal mouse spinal cord stained for nuclei of cells. And what you can see in the spinal cord of even a neonatal mouse, you've got tons and tons of neurons. They all look the same as one another. Okay, so while it may be possible to record from a single cell in the spinal cord that's rhythmically active during walking, find out where its axon projects to and get an idea of what its function might be during locomotion, okay, the chances that you're ever going to run into a cell with the same function as the one that you just recorded from are extremely slim based on the fact that you're dealing with a recording looking like this. Again, just as Rob said, a major step forward, or what was seen as a major step forward in the study of central pattern generators was this molecular dissection, genetic dissection of the mammalian spinal cord, which really started coming about maybe, let's say, 10 or 12 years ago. Okay, in which genes and transcription factors were discovered, which allowed the spinal cord in the embryonic stages to be divided into about 11 populations of cells. Okay, all of the cells belong to each population genetically similar to other cells in that same population, genetically different as cells in the other populations. Okay? So in the postnatal spinal cord, which I have over here on the right, you can see a distribution of neurons, each with neuron that I've illustrated here the same color, should theoretically have the same genetic background as all of the other ones in that same color. And if you go on the fact that genes determine cell properties and ultimately cell fate and function, you could take the step forward and say there's a chance that all of these cells expressing gene X, so all of the green cells, have one function during walking, all of the blue cells have another function during walking, etc. Okay, the really nice thing about using a molecular approach to study neural circuits, and the locomotor CTG in particular, is that you can use genetic techniques and molecular tricks to cause all of the members of a given population to express reporter proteins. Okay? So they allow you to visualize all of the members of a given population. So here we've got one population in red, here we've got another population in blue, here we've got another genetically distinct population, two more than them here in black and white. Okay, so we can now see cells that are genetically similar, and we can study each of these populations directly. Okay, so instead of randomly going into the spinal cord and reporting through cells, we can now focus our study, let's say, on B0 cells or on B1 cells, each of these populations. Okay, and that's what we do in my laboratory. Okay, and we use two basic techniques in which to study these populations. Okay, the first of which is known as effective locomotor preparation. So this is kind of a hybrid of that first video that I showed you where you had the spinal cord isolated in a dish and the legs kind of walking basically. Okay, so what we do is we dissect out the spinal cord of a neonatal mouse, okay, put it in the dish, and we can record, we don't leave the legs attached. Okay, what we do is we record from motor axons that travel out to the hindline muscles. Okay, so it's fictive locomotion because you're basically reporting, as you can see here, the neural correlates of walking activity. And when you've got activity on the left-hand side, okay, you can see the right-hand side is silent in a center. So you get this alternation. This is known as fictive locomotion. The second technique we use is one which allows us to record from these genetically labeled cells in the spinal cord. So what we do is we basically take the spinal cord out, we use fictive locomotor preparation, but like I said before, since all of the cells that we're most interested in are located by the central canal, we take off the top half, the dorsal half of the spinal cord, we put the spinal cord in a dish, okay, we can then use a microscope to focus in on the spinal cord. Here's the central canal right here, okay. So we focus in, we can see single neurons, some of them will express those reporter proteins that I talked about, for instance, green fluorescent protein, and we can record from single neurons that belong to one of those genetically defined populations. Okay, so here again, I've got the outline of the spinal cord with the different genetically defined populations. Like I said earlier, the key components of the locomotor CTG are located ventromedially in the general region of the central canal. Okay, today I'm going to talk very briefly about studies that we've done on two of these populations. First, the V0 cells, and then also the DI6 cells. Okay, so the V0 cells, okay, we recorded from these cells, and what we found, okay, so here's our spinal cord. We're recording the fictive locomotor activity. It's shown down here. When we make intracellular recordings from single V0 cells, what we find is that these cells are rhythmically active. Okay, and you can see that they're out of phase. Okay, so when they are active, okay, the motor axons on the contralateral side of the spinal cord is silent. Okay, and vice versa. When this is active, these cells are silent. Okay, we did anatomical studies on these cells, and we showed that these cells here, we've got a kind of cartoon of the spinal cord. Okay, this is a representative of the V0 neuron, and what we showed was that they cross the midline and connect onto the motor neurons on the opposite side of the spinal cord. And finally, the most striking result with these V0 cells, when we recorded fictive locomotion in the wild-type animal, okay, so this is an animal with all the cell populations intact, when we record on the left and right side, okay, you should see alternation. Okay, so when left flexor motor neurons are active, right flexor motor neurons are inactive, that's exactly what we see. Activity, inactivity, activity, inactivity. When this is active, this is silent. But when we get rid of the B0 cells, what happens is you have a breakdown in this alternation between the left and right side. Okay, in some cases you get what looks like pretty much appropriate left-right coordination, okay, whereas in other cases you get complete coercing on the left and right side of the spinal cord. So based on these studies, we came to the conclusion that the B0 cells are responsible for coordinating left-right alternation during walking. Okay, very briefly, the second population of cells that I have studied over the last few years and that we're continuing to study in the lab are these DI6 cells. Okay, so these originate dorsally, but they migrate ventrally, take up positions close to the central canal. We did many of the same studies that we did on the B0 cells with these DI6s. What you can see, these are also rhythmically active during locomotion, figure locomotion. The interesting thing about these cells, though, is when we synaptically isolate them. Okay, so we take away all of the inhibitory and extensatory inputs into these cells. You can see that they still remain rhythmically active. Okay, so these still burst rhythmically when they're synaptically isolated. Okay, so we basically came to the conclusion that they have some sort of intrinsic oscillatory property to them. Okay, the final thing that we did with these cells, or that we have done with them, we're not finished with them, is look at them, basically look at where they're located and did some axonal tracing. And what we found, here we've got a photomicrograph taken from one side of the spinal cord. This is the central canal. You can see that they're located in clusters close to the central canal. So we've got 4 of them here. We've got 3 or 4 of them up here. They're located very close to one another. And here we show that they're actually synaptically connected to one another. Okay, so this is the central canal. This is just sections taken, right, kind of serial sections from the spinal cord, 1, 2, 3, 4. Here are 2 DI6 neurons. You can see there's an axon connecting these 2 cells. Certainly seems to make a synapse onto the other DI6 cell. Okay, so what we're doing going forward with this population is we're trying to, this is very preliminary data right here. We're trying to find out the other cells that these DI6 neurons make synaptic contacts onto.